Yes, that's an important announcement. So everybody who's online now, we're actually going to record this meeting so we can also view it back on the ISACB website. So good afternoon, everybody, and welcome to our next edition of the ISACB Young Investigators webinars. First of all, happy 2025, and I think we can look back at a very nice and productive 2024. Not only did we have a very interactive meeting in Vienna, but we also had our first diversity and inclusion webinar. And this webinar was also given by Geerte Slappendel, our speaker of today. And in this webinar, we focused more on how to be an inclusive researcher. So today we're going to follow up with part 2, and that is going to be more about how to perform inclusive research. And because I think this is nice if we're going to have it more interactive, if you want along the way and you have any questions, please raise your hand. Geerte will be able to see that, and then when you have a question, when it's raised, just directly discuss it with us. Okay, so I'm going to give the floor now to Geerte, and we're really looking forward to hearing what you have for us in store today. Thank you.
All right, and welcome back for anyone who also was here for the previous session, and nice to see you for anyone who's here for the first one and missed the previous one. As Merle said last time, I mostly talked about diversity and inclusion in research teams and among researchers. And the other part, I think, of diversity and inclusion in research is in doing your research. And I want to take a brief moment to mention one of the reasons that I was able to pull this together relatively easily, which is NextGen, which is an EU-funded project that I'm part of, that we're coordinating from UMC Utrecht. I've got a quick overview of partners here and all the formal statements. What we're doing in NextGen is we have a 4-year project where we have 21 partners from anywhere from all over Europe to the US, collaborating to create, and I'm going to read this because it's too far out of my own comfort zone, to build tools to enable portable, multimodal, multi-omic research for personalized cardiovascular medicine. So what they're doing is pulling together all the databases, biobanks, etc., that we have in various participating organizations, putting together all kinds of different types of data that are stored in different kinds of formats and gathered with different kinds of tools, and try to pull them all together with federated learning systems so you can set up personalized risk predictions for cardiovascular medicine. And it's a huge undertaking, and there's a lot that goes into it. And one of the reasons I ended up here giving this talk is that I was originally involved here as a project manager, helping to set up all the paperwork with the European Union, etc. And I got into a conversation with one of the coordinators about diversity and inclusion and about the importance of addressing these issues. And he said, well, can you please help? Because you are so right. We need to work on this, but where do we even start? Because one of the things I find is that particularly with AI and machine learning coming up, there are so many issues that a lot of people are kind of vaguely aware of the fact that there are issues, and everyone is still struggling with how to deal with them. I'm not focusing today on AI and machine learning. I'm really going to broadly talk about biomedicine and biomedical research and what's going on there. Because the basics for me is that excellent research is diverse and inclusive research. And I often do. I do a lot of talks around, well, this is what your funders will ask, and this is what requirements are and regulations are, and this is what you need to do to get the money. But the point isn't for me to get the money. The point is that this is how you do excellent research, and that this is a moral obligation, because in the end, the money is a means to an end. This is the end. I'm going to address briefly here matters of definitions. A lot of the discussion around diversity and inclusion in research in Europe tends to focus on sex and gender. And that's kind of different if you're in different places. The US tends to focus a lot more on race. Canada tends to focus a lot more on native populations. In Europe, a lot of these conversations start around sex and gender. And you'll notice I'm saying sex and gender because they overlap, but they're not the same. If we're talking about sex, we're talking about biology. We're talking chromosomes, we're talking hormones, we're talking physical characteristics. If we're talking about gender, we're talking about the roles we take on in life, the responsibilities we take towards others, and how we present ourselves towards others. And the overlap is large, but not perfect. Gender is also culturally defined, so it shifts, and you see a lot of it shifting in the current time period when it comes to what do we think is part of being a man or being a woman, and you see a lot of clashes around that. But it's important to know that both can influence biomedical processes and both can influence your health. And often you won't be able to pinpoint which of the two is causing an effect, but it's important to be aware that these are different pathways. Diversity is more than just sex and gender, though. It's sex and gender, it's ethnicity and race, and I'm going to touch on that a bit more in a moment. It's social class, it's sexual orientation, it's age and religion and physical ability, neurodiversity and mental health, cultural backgrounds. And when it comes to health, all of these have their effects. When it comes to race, I want to be explicit for a moment because this gets sensitive and it gets complicated. Because it tends to be very easy to talk about race, but it's really hard to define what you're actually talking about. Because biologically speaking, there is one human race. There are not enough genetic differences between different human populations to justify speaking about different races. But sociologically, it is definitely a very real concept because it affects how people are treated, it affects their daily lives. There is a concept called minority stress, which relates to people who experience discrimination having a constant level of chronic stress that has very real effects on their health. And it gets very complex to really pick these things apart, but I find it very important that people are aware that there is a very real thing called race. It is in large part not based on biology, but on culture and social positioning. And that means that if we're talking about race, we're talking about a lot of different things. We're talking about ancestry and genetics. We're talking about ethnicity and cultural identity, including all the lifestyle differences that might entail. We're talking about physical characteristics because due to the degree of segregation we do still see between different subpopulations, there are differences in physical characteristics because we're not perfectly blended. there's social perception, there's migration status, and all of those can have effects on medical status. Flanagan et al. Have a paper out with information and guidelines for, I believe it was JAMA, if you're doing reporting on ethnicity and race, including how do you define the groups, what terminology, what do you report. Merle, is that already an answer to?
Yes, partially, because I was kind of now wondering, I always kind of took it for granted, but then how can you be both sensitive but also specific enough if you want to include groups of people? I think one of the examples is that previously, when determining renal function, you would have like a part in the equation that would denote black race. That is something that is now already out, because one of the ideas was that kidney function in black race was different than in white. But if you still find that physiology, same as what you discussed last time, is different, then how can you denote who is included or not? Seems
I
very
mean,
complicated.
it's really, really tricky, and I think that really diving into that, you know, is not even something that I would be able to fully do in an hour or 45 minutes, even if that's what I was trying to do. I think that a lot of the important, I think one of the main things to consider is actually to generally try and include people broadly, because even if you're not splitting it apart by subgroup, because splitting apart the subgroups, my mother-in-law is Black Surinamese. My father-in-law is white. My parents and I are both white. How do you classify my kids? There is no way of knowing, because you have no way of knowing which genetic markers they did or did not pick up. But if you make sure that your overall population of people that you're including includes a wide range of people from different backgrounds, then your data is going to account for this full range of diversity that you see within people. And that means that, particularly if you're setting up larger scale models, your model is able to account for this full range of diversity rather than being tailored towards one specific group. But yeah, picking apart who goes into what group is complex and not easily answerable, and will very much depend on exactly what it is you're researching, what pathways are involved, and that's what differences it is you're looking for. And it can be very relevant. I know that if you're doing genetic research, one of the things they tend to do is you have certain genes that tend to inherit together, that tend to be linked. And I know that there is a particular study where they found that for a particular gene that they tend to track in that way, within the Han Chinese population, these two genes don't actually co-inherit. So if you're trying to do genetic testing in someone with a Han Chinese background, and you're doing the usual process of, oh, we know that they have this gene, so we can assume that they also carry the target gene, you're going to be wrong, because for that particular population, the two genes aren't linked. So it can have very real effects in very unpredictable ways. Then to take it back to excellent research, I argue a lot that biomedical research is inherently applied research. And you can, move along the scale a bit in how fundamental it is and how theoretical. But in the end, because biomedical research is always about humans and about how the human system functions, you're not doing anything particularly useful if you're doing research that you're disconnecting from the rest of the system, because it uses its effectiveness. And that means that if you really want to do excellent biomedical research, you need to know not just if something works, but under which conditions. You need to know how it works. In a lot of cases, I think that having this diverse group in your research and knowing in which groups differences show up will also tell you so much about how your differences actually come to be. Because if a difference between men and women shows up, or a difference shows up between people with a lighter and a darker skin color, then that says something about the processes you're studying. So this is my basic argument. Excellent research, excellent biomedical research in particular, because it's applied research, because it studies systems that cannot be isolated from the total, needs to be diverse and inclusive to really be excellent. As I said, I work with NextGen, which means that as I was setting up the workshop for them, I did a lot of work in finding papers around cardiovascular health to look for places where this becomes relevant and where issues start popping up. So what I'm going to start off with here is just start throwing some examples at you of things that have been found. And I think this is one of the more straightforward ones. This is a review where they looked at gender aspects in cardiovascular pharmacology. And this table shows you an overview of, well, in beta blockers, we see similar benefits, but women get a lot more side effects. So you might need to spend some more time looking at how do you dose this so that you balance the beneficial effects versus the side effects well. For the digitalis, you see higher mortality in women. So you might want to be more cautious in how you use that. And one of the things to keep in mind here is that some of this will also be age dependent, because one of the things that tends to affect the processing of medication is the presence of estrogens and progesterone. So if you see differences between men and women, it's worth looking into whether you see those differences at all ages. I know that they've done studies on antipsychotics, where they found that in premenopausal women, they needed to substantially lower the dosage of the medication to get the same blood level of medication as in men. But for postmenopausal women, they needed to give women the same dosage of medication to get the same blood levels. So again, fairly straightforward, but something that's very important if you're doing anything that is going towards actual treatment to be aware of. I suspect that a lot of you are probably aware of the history of doing medical research only on men. And that means that a lot of the time, there's a lot here that we don't know. It gets even more complicated when it comes to how do you deal with this during breastfeeding or during pregnancy. And there are databases at this point where they keep track, because obviously not all women have the luxury of being able to go off their medication for a pregnancy. So a lot of medication does get taken during pregnancy, and they do have databases these days where they keep track of what happens then in terms of side effects in these women who take this medication because they have to, so that they can better advise women on how to do this safely. Then heart failure algorithms. This one is straight up sex. What this paper did is they pulled together a lot of, well, they ended up with 16 different AI models for prediction of cardiac disease. And what they did is they retrained it on their own data and then split it up to check if we're separating this between men and women, how well does this model perform? And I honestly find it somewhat scary that essentially the norm is that women are underdiagnosed and men are overdiagnosed. Because that's 13 out of 16 experiments where they find that they're over-predicting disease in men and under-predicting in women. And I think that the thing to keep in mind here as well is the fact that this conversation tends to focus on the fact that we're underdiagnosing women, and that's an issue, and that's a huge issue. But in this case, what you're seeing is that by not pulling this apart and not looking into these differences, we're also overdiagnosing these men. And I don't think that any one of us would really want to get a diagnosis that's not justified any more than we want to be missed. Then this one, and this one I actually just found the other week because I was talking with a researcher about a study he's planning to do. There are ethnic and socioeconomic differences in how effective these algorithms are as well. And this one, they looked specifically in the Netherlands. They used the SCORE2 model, and they found that overall, in low socioeconomic subgroups, they're already under-predicting. But then if you're looking specifically at the subgroup of Surinamese people in the Netherlands, you get 2.5 times the number of actual events compared to what's predicted for Surinamese men. And that is a huge gap. And I know that there's a researcher in-house who's working on setting up a study to see how to improve this. Then, in the interest of not going too far out of my lane, I'm going to have someone with actual expertise on what I'm doing or what's happening here. Yeah. Merle? Seeing your hand.
Yes. So, of course, there's a problem in the model because probably events in low socioeconomic groups like Surinamese men are not included in making a model. But I'm kind of wondering where then this discrepancy comes from. So partially maybe in that low socioeconomic people tend to be more, let's say, afraid to go to a GP, or are there what would be the things underlying this problem?
That's one of the things that the follow-up study is hopefully looking into. But I think that there is a factor of stress.
Right.
I mean, when it comes to cardiac health, stress is always a factor, and having a lower income, not having the same level of confidence that you will be able to pay your bills, or you will be able to buy a new car if you need one, or etc. That probably has an effect. There are effects probably of health literacy, of how easily can you afford to pay your copay to go for a doctor, take time off to go to a doctor. What time and energy and money do you have to go work out, etc. The question is, and I don't know the model well enough, because some of these, I mean, there are effects of smoking, for example, as well. But I would guess that if you're setting up a model to predict cardiac disease, that probably goes into the model. One of the things that they probably ask to set up this risk prediction is, do you smoke? But yeah, and one of the things that the researcher who's working on this was actually arguing is that it's very hard to pick apart what is an effect of socioeconomic status, what is an effect of ethnicity. And then there's also the question of, well, how do you define your ethnic groups? As I said, where in this model do you put my husband or my kids? Because I think this study looked at country of birth to determine ethnicity, which means that you're excluding any second-generation migrants. But I think that there are probably effects there that do work out over multiple generations post-migration. But yeah, puzzling that out, again, is tricky.
The lack of inclusion of minorities in clinical biomedical research has real-life consequences. There's a genetic test that can predict whether you're at risk for an enlarged heart. A lot of student athletes get that test because it's part of a physical exam. And many African Americans were overdiagnosed with this disease, erroneously diagnosed. They were told to stop doing any strenuous exercise. Maybe we should put you on antiarrhythmic. Maybe we should put a defibrillator in you. Maybe we should test all your family. And the New England Journal paper showed that many of the diagnoses were misdiagnoses. When they went and looked back at the genetic test, the test was really developed in whites, and the gene frequencies differ in blacks and does not have the same effect in blacks. And so the test was biased and misdiagnosing lots of African American patients.
This is a fairly short clip. It's interesting to look into the rest of it as well if you have the time at some point.
I don't.
Go ahead and stick to this particular example right here, because this is the paper that he just showed. Again, multiple patients, all of whom were African or unspecified ancestry, received positive reports with variants misclassified as pathogenic on the basis of the understanding at the time of testing. Subsequently, all reported variants were recognized as benign. And I highlighted this particular bit here. Inclusion of even small numbers of Black Americans in control cohorts probably would have prevented these misclassifications. And this, Merle, I think, is the point that we were discussing earlier, that even if you're not splitting out these different ethnic subgroups and coming with targeted recommendations for each group, just having them included so that you have this full range of what is normal included in your data makes a huge difference in how useful your eventual results are. Then I was talking earlier about sex versus gender, and I actually ended up in a bit of an argument at a certain point within NextGen, because one of the researchers there was arguing, well, we're just dealing with sex. Gender is not relevant. It is. It is. It is relevant partially because of effects like this. There are gender differences that might not fully align with biological sex in how we deal with stress, in what stress we experience. There are effects of the stress of having to take on the mental load for your household, taking on the care for family members who might need extra support, that have very real effects on health outcomes. there's also this fun bit when it comes to clinical care, because I think we all know that women tend to get under-included in trials. And there's a lot going on there, and that partially has to do with who we approach, where we approach them, but it also has to do with the fact that women tend to be more risk averse. So women are a lot more cautious about saying yes to these things, and then researchers come to the conclusion, oh, but if I approach a man for this study, he's more likely to say yes. So whether intentionally or not, it's very easy to just start approaching more men because it gets rewarded a lot more. And then it all stacks on top of each other. One of the solutions I hear being discussed is, well, let's use healthcare data. Let's just use data from their patient files. And in theory, that would be a very helpful solution. But here's the problem. If you have patients presenting in the emergency department with chest pain, women are less likely to be triaged as emergent. They're less likely to get an ECG. They're less likely to be admitted into the hospital. So that means that if you're looking at patient data, you're going to have less data on the women. And there's a couple of papers that we have out on a cohort that we run within UMC Utrecht. And what they do is they have set up a learning healthcare system. And they're trying to set this up so that they don't have to do specific informed consent, but can work with an opt-out. Because one of the things that they actually find is that, one, women give less informed consent, but also there is a gap between the data that gets recorded about men and about women. Women receive less protocolized care, so we have less data on the standard measurements that are supposed to be taken, and this is a difference of like 30%. You get 30% more missing data on stuff that should be routinely monitored for women compared to men. So even if you're using these healthcare data to get around the issue of whether or not people are opting into your study and giving informed consent for a trial, you're still going to have less data on the women because they're not getting the same level of care. And the follow-up that we're currently working on, and I hope to get a paper on that submitted in a few months, is that you always have to have an opt-out in place still for the secondary use of healthcare data. And again, who uses this opt-out is not equally distributed across different groups. Women, and particularly highly educated women, are more likely to make use of the opt-out. And when they make use of the opt-out, they also make use of the opt-out for their kids. So now what? And I've got a few suggestions and ideas here on things you can do. One of the things I want to point out up front is I spent a lot of time talking about eating elephants. You do not eat an elephant by trying to stuff the whole thing down your throat at once. You take small bites and you save things for tomorrow, and you bring a lot of people, and not everyone does everything. So I have things that you can do, but be aware that you don't have to do everything every single time, because doing a little bit is better than doing nothing. When you're planning your study upfront, have a look at the literature. There is actually a database specifically for research on gender differences that you can look and see whether there's anything in there about what it is that you're studying. But even just, looking into whatever your keywords are and adding gender or sex will often get you a start. And that gives you an idea of what should you be looking at, what should you be considering. Include target groups in planning. One of the things I suggested for the study I was talking about earlier about adjusting this score 2 algorithm for different ethnicities is to, reach out to Surinamese population, reach out to the Moroccan population and talk to people there about how you can get them involved in these things and what it is that they need from your studies and what issues they think are at play. Because a lot of the time, people have lived experience that is going to be very useful in helping you determine what might be going on. Unless you have a reason not to include diverse groups, and in some cases, I mean, I'm not arguing that you should be studying prostate cancer in women. So there are exceptions. There are reasons why you might choose to do a study only in women or only in a specific population, but only do that if you have a reason why you're doing that and be clear about that. And know your limitations. Sometimes you just know upfront that something isn't going to happen, and then it's better to just acknowledge, well, ideally, it would be good to do this for these and these reasons. It's not going to happen, so we're just going to have to work around it, acknowledge that it's a limitation, but be aware of that. When it comes to inclusion, and I phrased a lot of this around, working with patients and working in clinical studies in that sense. But for all research stages, also if you're doing preclinical work, include at least both men and women. If you're doing work with cell lines or work with animals, try and have both male and female animals, both male and female cell lines, ideally have cell lines from multiple ethnic backgrounds, and make sure that you report on that when you're publishing your study. Because I have heard a few too many people say, well, I'm doing cell studies, sex doesn't matter. Your cells have these sex genes in them, encoded in them. So it is absolutely possible that even at a cell level, you're going to see these effects, and you won't know until you try. That said, I am well aware of the fact that this is not necessarily a cheap and easy proposition. But for some funders, such as for EU funding, this is absolutely something where you could just request the extra funding. When it comes to getting minorities or minoritized populations participating in your study, have a look at why they're not participating, because often it's not just that they haven't been asked. It can be, if you're trying to get women to participate, they often have a lot more care responsibilities. They need to plan things around taking care of their kids, taking care of their parents. But then who's going to cook dinner? See if you can help out there. Can you offer childcare if they cannot leave their kids at home? Can you offer a ticket for a meal, or have a look at what responsibilities might be keeping them from participating and see if you can offer something to make that easier on them. There tends to be a lack of trust, and one of the things that actually helps there is if you can show that you have a team where you have people from different populations in your team. Studies with female PIs tend to get more women enrolled. And there are likely similar issues at play if you're trying to include people from ethnic minorities. Seeing that people like them are involved in your study makes it easier for them to trust that they will be treated well. And make no mistake, particularly ethnic minorities have been very hard done by previous researchers, and people are very well aware of that risk, of that history. So they will be hesitant, and they will be need to be given a reason to believe that you're not going to do anything like that for them. Again, women tend to be more risk averse. They just tend to, particularly when they're under stress, like, oh, say they're in the hospital being treated for a disease that is going to affect their lives long term. They tend to be very cautious. They don't want to take any risks. So you will need to see how you can offer them some reassurance that they're not taking any unnecessary risks by participating in your study, more so than men. We were talking about socioeconomics, that also relates to language skills, that relates to health literacy. If you're having migrant populations, do you have information for them in different languages? Can you offer a translator? Things like that. And with language, don't underestimate the number of people who do not read well, because there are a lot more people who have really limited literacy. So have your information for your patients as simple as possible, with straightforward language, not too complex. See how you can simplify. I also cited location. I suspect a lot of people work in academic hospitals. And the population we get in academic hospitals is very easy to approach for us. So it's really easy to think, well, I'm just going to pull from our own patient pool. But there are huge differences between groups in how likely you are to end up at an academic hospital. It's, do you go to the doctor to begin with? Does your family doctor take you seriously? Do you know that you can push for a referral to a hospital? Do you know that you can say, well, I don't find this level of permanent symptoms to be acceptable, so I want to be referred even further? Are you listened to when you ask that? So if you're wanting to have a diverse population included in your study, you will want to at least, include regional hospitals that serve a broader population, or see if you can reach out to family physicians to see if you can include their patients. But not everyone makes it to the academic hospital. But also when it comes to heart problems particularly, be aware that women have different kinds of heart problems. So women with heart problems are less likely to, say, get a bypass surgery. So if you're doing a study on secondary heart prevention and you're recruiting your patients by looking at the OR rosters and checking who is coming in for a bypass, you're not going to get a lot of women in your study, because you're not looking for your patients in the place where the women are. So that's a different kind of location. As I mentioned, be aware of the gaps in healthcare data. If you're making secondary use of healthcare data, and build bridges with the affected communities where possible. When it comes to your analysis and reporting, I think that the garbage in is garbage out is probably familiar to many of you, but this also means that if you're making use of healthcare data and you know that particular groups are often underdiagnosed, then your algorithm is going to replicate that underdiagnosis unless you do something about it. So take into consideration, what is the data that I'm putting in? What are the biases inherent in that data? Is there anything I can do about it? Stratify analysis, and I know the standard answer there is, I don't have the power. I don't care. Because the thing is that on the one hand, as I mentioned at the bottom, there's a difference between can you get statistical significance or is there clinical significance? And I think that there are absolutely situations where you will see a clinically significant difference that doesn't reach statistical significance because of power issues. And then you can discuss in your paper, hey, this difference didn't reach statistical significance, but if it's real, it's highly clinically significant, so there needs to be follow-up on this. And if we make a common practice to always report stratified results, then if a researcher shows up and says, well, I want to do a meta-analysis to see if there are real sex or gender differences here, they can do that. But I know that we have a researcher within UMC Utrecht who wanted to do a study like that, who wanted to pull together data from different studies to check for sex and gender differences. But just about none of the papers she could find reported stratified results. And you can do this in supplementary materials. You don't have to put it into your paper. But if you make it available, then others can find it and others can then pull together all this stuff from the different papers to get answers that you might not be able to get within your own study. Similar reasoning, discuss the knowledge gaps. If you have a situation where you go, well, I wasn't able to stratify, or I wasn't able to get enough women or enough different people. Again, if we all start making a habit out of saying, well, this is an issue and this needs to be addressed, then the next person who is trying to write a proposal for a grant has the references to get the money to fix the issue. But they need to have the literature base to argue for that. And then this one, and this is very recent, as you can see, December of last year. We've traditionally excluded women from clinical trials because they are so hormonally unstable, it messes with our results, and it's too complex. But if this messes with your results so much, then why are we not looking at how and why this messes with our results? Because if it messes with our results, then it must be relevant to our results. What they found was that they had differences in the effectiveness of chemo treatments between different mice, and they could not explain why are these mice routinely responding so much better than those mice. And it turns out that it was because of the phase of the menstrual cycle of the mice when they were giving the chemo. Turns out that if you start chemotherapy during menstruation versus at some other point in the cycle makes a difference for how effective your chemotherapy is. Where might this be at play within cardiac health? Where might this actually, because one of the things that I've been reading is that estrogens tend to be protective when it comes to heart health. So how can we make use of that in our treatments and in our diagnostics? Then, to come full circle, This was an article on the Uncode Institute's website about the paper I just showed. And what I find hilarious is this is a male researcher speaking. It suggested that measurements were perhaps taken during different cycle stages. At first, I didn't pay much attention. I didn't think the cycle would greatly affect the cells. But when I showed the results to my wife, she said, "That makes sense, right? Of course there could be an effect." Again, at the bottom, at medical conferences, I noticed that these findings evoke different reactions. Some colleagues, especially men, react skeptically, as I did initially. But many women agree and recognize the possible influence of cycle. Getting diverse researchers to the table makes it easier to get these things recognized and to get these things acknowledged. Because having these different lived experiences and these different views on how things work makes a difference for what it is you pick up in your results, what you're seeing in your data, what questions you're asking. And finally, and I'm gonna stop sharing here, I'll send these slides to Merle so she can distribute a few resources and places where you can find more information.
That's great. Thank you very much, Geerte. I was looking if I could find an emoji. Yes, an emoji like this. I also already looked up the first article. I thought it was Flanagan or something. I thought it was super interesting, and I also already posted the link in the chat. So I will make sure to distribute this to everybody. Thank you very much. I think it's really thought-provoking, and I probably have to think about it a bit longer. Maybe there are questions from the audience, and when they think about questions, I already have one. And the first question that came to mind here was, is this specific? Well, the last slide you talked about chemotherapy. Is this specific for cardiovascular biology, or is this equally bad in other disciplines of medicine as well?
I mean, I think that cardiovascular biology at the moment is doing relatively well. There are a lot of issues, but there are also quite a few researchers who have particularly women's hearts as a focus area at the moment. And that is bringing a level of awareness that's not necessarily present in other fields. But it's an issue across the board of not even just biomedical research, but I think all kinds of research. It's an effect of the culture that we've, collectively been raised and trained in.
Yeah. And is it also, you probably have a better view, like very different across the continents? Like, is the focus here different than, for instance, in North America, or if you don't know, then also I was just wondering. So for me, some of the things sound relatable, but I was wondering if you live somewhere else, maybe the issues are very differently.
I mean, I think that part of the issue here is that there is a fairly limited range of geographical areas where we tend to fully take seriously the research. And that kind of limits the answers here. As I said, in the US, there tends to be more focus on ethnic differences, on racial differences than there is here. Whereas in Europe, we tend to focus more on sex and gender. I think that there are quite a few people in what they tend to call the global South who are doing a lot more work around, how does this translate to the people who live in our area, to our culture. So I do think that there are regional differences there in what differences do we consider important and what differences do we look at.
Right. Yes. Okay, so are there any other questions online? Maybe if you don't have your camera on or have trouble with your microphone, you can also type in the chat. Just speak up otherwise.
So thank you again for a wonderful presentation. The end of your presentation really struck me about the differences in how PIs with diverse backgrounds might view certain results and be dismissive of certain things. So I'm curious, like obviously having diverse PIs is going to contribute to this type of research, but for those that are less diverse, do you think that the change in their perspectives and how they interpret results can, like, how does that change start? And is, like, you know, as a professor that's also an educator, can it start in the classroom? Like, do you see that maybe a new generation of PIs, whether they're diverse or not, would also be more sensitive to these findings? And like, do you think that should be mindfully like incorporated into lectures? And
I tend to actually be really allergic to the suggestion that you would only be able to do these things if you're part of a particular subgroup. I know white male researchers who are very aware of these issues and very actively working on it. So yeah, I think that there's a lot that can be done here, regardless of what your particular background is, in just, making sure that you listen when others talk. When you keep an ear out for what experiences others have, create a research group where people feel free to say, well, hey, this doesn't actually match with my experience or with how the people in my community live their lives. Having that openness to think about those things is not something that's tied to any particular identity, and that's something that everyone can do. And I know that within UMC Utrecht, we're currently very active when it comes to our medical education, in working on, well, what patients do we show when we offer case studies? What information do we give to these new doctors we're training about how things look in different skin tones? I mean, when it comes to cardiac health, one of the things that you tend to look for is pallor. Like, does someone seem pale? I know I've had a conversation with someone who was a person of color himself, because we were at a party. My husband's grandmother was, not feeling well. And I said, well, she's looking all pale. And someone said, no, she's dark skinned. She can't be pale. I see the actual difference between her usual skin color and what she's looking like now. She is pale. It's relative to a different standard than you might be used to. But she is absolutely pale and not looking well. And I can see that in her skin color. But since, particularly when it comes to white people and white highly educated people, we're so used to having an environment that's entirely comprised of white people, we never get used to what things might look like on a different skin color or in a different ethnicity. So yeah, that's absolutely something where we can train people better. If you're interested in how things like that might look, there is someone, and he's active on LinkedIn, and he's called Chidibere Ibe. And I can type that up in the chat because I am aware that that is not.
thank you. Yeah, I appreciate that.
What he does is he does medical illustrations in different skin colors. So for a huge range of medical illustrations, he creates illustrations where he compares how this looks on a dark versus a medium versus a light skin color.
I've
And
seen some of his illustrations. I think it was on pregnancy. It was actually really beautiful as well.
he does really beautiful illustrations and is very deliberate about, showing a diverse range of what people look like in his illustrations.
That's awesome. Thank you for sharing that.
Thank you. Are there any more questions or maybe just remarks in general? For me, as I said, I think more food for thought. And I also like the idea that you can be the one starting the discussion, even though it's maybe not your ethnicity or not your background, that you at least you have the power to start the change or start the discussion on maybe we should do differently. So I think that's a powerful message.
Yeah, and it's one of those things where we're not going to change anything overnight, but if everyone starts doing little bits, then you can slowly get things moving in the right direction.
Yeah, I think that's a great way to end as well the webinar. And we're gonna write, I think, for the newsletter a small piece of summary, and I think we should start or end the message with this, like if we as the researchers of tomorrow and a bit of today start with small things that we can change, then over time it will be better. But there are some great initiatives already, but there's also still a lot of work to be done. That's very clear. Perfect. Okay, Geerte, again, I want to thank you for now two times a webinar for the ISACB Young Investigators. We're going to make sure that we post this online, and we know also from our community that people tend to watch it back online. So I will get back for another, I think, contract to have it appear online. And later, somewhere this month, I will come and visit you at the UMC to say another thank you in person, which I think would be really nice to see each other again. And for now, the Young Investigators are coming up with a new series of webinars for the 2025 edition. So please keep an eye on both the e-circulator and the website, and then you will see us again. Thank you, everybody, for attending. And Geerte, again, thank you for your talk and the exciting content. And we will see each other again. Thank you very much.
Thanks.
Thank
Bye.
you.
Thank you.
Thank you.